It's really about, like, if you're looking at like omics or genomics or transcriptomics, people think, oh, you know, all these sequencing things are the same, and what I'm going to tell you is that they're really not the same. And so there are different sequencers out there, there's different ways of doing library construction, and I'm going to talk about some other things like quality scores and computational pipelines. So there are different sequencers, and all the sequencers I'm going to talk about, they do exist still. So this one's called Helicose or Sequel, one of my favorites of all time. We do have an instrument here at MGH that is in my lab, but they are a company at Woburn. They used to be in Cambridge, and they used to be called Helicose, and they had this machine that was what we call a single molecule sequencer. I'm going to explain what a single molecule sequencer is, because this type of technology is like the basis of all the next gen sequencers. But it was a very simple idea in that what you would do is on your flow cell, which is like either glass, optical glass, or plastic, you actually would put oligo DTs on the sequencing flow cell. You just got a bunch of T's. And then the idea was, at that time, was like, oh, you could flow RNA onto the flow cell and it would get caught because they've got this poly A tail. And then you could do cDNA synthesis with superscript 3 or something, create a cDNA, and then you RNase away the RNA. And then what you have is a piece of cDNA stuck to the flow cell. And then you can actually use terminal transferase, and terminal transferase only puts things on the end. And you can add a bunch of G's, or G's here. And then the next thing you can do is use a poly-C primer, and there's not poly-C in your genome. So it would only prime to this end, and then you would initiate sequencing by synthesis. So you would add your A, your T, your C, your G. And each one of these A, T's, or C, or G's was a different floor, a different color. And so what you could do then, and this is how all these sequencers work, is you could take images of A's, T's, and C's, and G's as you added it. And you would add a color, it would light up, take a picture, kill the color, add the next base. There may be no color, so you don't call that. And so you can actually take all those images and stack them on top of each other because you know where their XY coordinates were, and have what we call massively parallel sequencing. Each one of these dots here, in this case, was a molecule of DNA being sequenced. So that's why they call it single molecule sequencing. So this machine is insane, right? Because you have to take a microscope that can actually image one molecule with all these molecules at the same time. So this machine actually weighed about 2,000 pounds because it had to not vibrate. But the concept of sequencing by synthesis and also sequencing in massive parallel is retained across all the major next-gen sequencers. And so we'll just focus on Illumina because Illumina did the same thing, but it was a little different. And the differences between Helicose and Illumina are important to know because there are certain things that Helicose can do that Illumina can, and certain things Illumina can do that Helicose can. And so the Illumina sequencer uses the same kind of idea. So it's a flow cell, okay, and each one of these is a channel or lane. I think they call them lanes. On each of these lanes, again, it's optical plastic or glass. In this case, it's plastic. You have all these what we call P5, P7 sequences. So there's not those T's. But on your library, you always put P5 or P7 on the end for library construction, which is just a sequence that is unique to the Illumina sequencer and not found in any other organism. Okay. So what happens is you make this DNA library and then you denature it, and then you hybridize it to the flow cell, just like what I showed you with the Helicose. And then you do this bridge amplification. So the other side of this P7, this side's P5, and it comes down, and I still don't understand exactly how it does this crazy thing, but it creates clones. And so these clones, or clusters, as they call it, so when we talk about clusters, they talk about this, becomes a clone of that original cDNA, or library, that you amplified, okay? And so when you do that, then you do the same thing that helicose did. You add the A, T, C, and G. Except when the A lights up, it's not just one strand, it's a cluster, it's a whole clone farm. And so imaging that is a lot easier. You don't need this microscope that has to try to image one molecule. You image a cluster. And so this is what made Illumina destroy everyone, is that they could sequence much faster because they didn't have to take a long time imaging. Because you could image, when the thing is bigger, it's just faster to image. Does that make sense? And so that's why, even though helicose had this really cool thing, Illumina won because it was just a lot faster. So helicose took about 8 days to run. You press the button and like, pray that the power would not go out. Illumina, took about the same amount of time, but you can sequence a lot more data. And then over time, they created their run times to be much shorter because they were able to sequence much faster. And so that's how the Illumina works, okay? So it's basically the same type of concept, but slightly different. So that's the HiSeq and the MiSeq, okay? And then some of you might be like, well, there's this NextSeq, that's the same. But it turns out that Illumina NextSeq is a little different. So remember, in helicose and HiSeq and MiSeq, you added 4 different colors. Okay, so you have to actually take 4 pictures for any given phase. Does that make sense? Here they just said, okay, let's just make 2 colors. So a green is a T, a red is a C, an A is the fused of a red and a green, and a G is no floor. No floor. Okay? That's kind of hard, right? But basically you can now just image 2 colors. You've cut your sequencing time in half. And that's why the NextSeq is so fast. But what's the main thing you think could happen with NextSeq that you probably have less of with the HiSeq or MiSeq? Anyone want to guess?
Calling G's is probably a little bit.
Calling G's is pretty ridiculous. And why do they pick G, you think? Because most of your genome is G depleted. Yeah, there's human genomes and mouse genomes, not as many G's. So. And there's not a lot of G's after each other. It's not usually, you don't see a lot of G. Okay, because high CG content leads to, anyway, so high CG content is very difficult for Illumina to do. But the one thing that you can do is you can, the error rate is actually higher too, because you're imaging two colors instead of 4 colors. So that's something to know, that NextSeq is not exactly the same as HiSeq or MiSeq. So some people are like, oh, I'm going to run some of my samples in HiSeq, some of my samples in NextSeq. You're a little different, so you might not want to do that. Okay, all right, so I just put the cost of how much these things are, but we can talk about that later. Another machine out there called PacBio. I think Illumina just bought them. So PacBio is a very different sequencer. It's sequencing by synthesis, but they actually put the polymerase at the bottom of their sort of sequencing flow cell. And so you actually image polymerization live. And so that's very cool, but that comes with pretty high error rates. So the error rate's about 11% per base. The way they get rid of that error is that they actually sequence the same strand like 1,000 times. And so the chance that you have that same base error 1,000 times at that same location at 11% is basically 0. So that's why they say we're accurate, because the consensus call is very good because we sequence the same thing. The problem with that is that they have low number of reads, only about 50,000 per run. But they could be really long reads, 10 to 15 kb. So this could be useful for like if you were like on Mars and you found alien life and you wanted to know what the genome kind of looked like because you didn't have the assembly. You would do this with maybe Illumina to sort of put everything in context. Remember, Illumina is like 300 base pairs basically. And so, what we used to do is we just snapshot shotgun align those things, so you would look for overlapping things. But when you can do a really long read, you know what it looks like in its total length. and then you can use Illumina in between to improve your accuracy. Does that make sense? So you need to have both pieces of information. So this is great for de novo assembly, and also things with high GC sequences like repetitive elements, which is something I've talked about for days. Anyway, so the other sequencer out there is called Torrent. This is one of the sequencers that is in the clinical realm, so competing with Illumina. And so this is now owned by Thermo Fisher. So this sequencer works, instead of by imaging, it detects protons. It's actually a really sensitive pH meter. And so every time you add an AAT or C or G, it would detect a proton. And so you can imagine that this is very fast. It's basically an electric type of signal. And so this sequencer is very fast, and that's why the clinical side likes it a lot. But it comes with, higher error rates. So, it has some advantages in speed and target sequencing, but it has some disadvantages in error. And this is like, I would say, it's no longer a unicorn because I've seen it now, called the Oxford Nanopore. So this sequencer was an interesting concept, actually theorized by physicists, like when I was an undergrad, which is now a long time ago, where you would actually make a pore, a nanopore, a protein pore, and you would actually use the fact that ACs and Ts and Gs have different impedance. If you have a current across this, an AC or T or G is going to be slightly different in the amount of current that goes past. Okay, and so you could theoretically sequence the entire genome like really fast, right? Because you're just waiting for this whole thing to just go through and it'd be really fast. The problem is that, the sensitivity of detecting an AT, C, or G through changes in current is not like super accurate. The error rate is fairly high, but the speed is really fast. And you can miniaturize this because it's not imaging, right? You don't have to have a microchip. So it's actually, the smallest one they have is one of these things that you can plug into your laptop. And so we have one of these upstairs, and it sits in the Haber lab right now. So it's really cool because you can do things very fast. You can potentially, envision like, oh, I'm going to fly to Africa and just go sequence some, random safari animals, and so highly portable too. So it's a pretty cool thing, and there's certain types of sequencing you can do that Illumina can't do very well. So one of them would be like native methyl-seq. So if you're interested in DNA methylation and don't want to do bisulfite, theoretically this would see differences between a methylated piece of DNA versus a non-methylated DNA because it's looking at differences in impedance. Okay, so it's pretty cool. So this is just a summary table, and I'm not going to go to this in big detail because most people here are probably doing Illumina. The error rate is about 0.1% per base, so it's 1 out of every 1,000 bases is wrong. There is amplification biases. You can't really sequence repetitive elements because you require PCR. So if you're interested in that, we can talk more about that. SQL is a single molecule sequencer, so it can sequence repeats. But you can sort of see there's a read length, and the DNA input that's actually a little outdated because there's these new amplification techniques that I'm not going to talk about. But my point in showing this is that not all these sequences are the same. They all exist. And people out there are using them for different applications for whatever their question is. So you can't really think about what can Illumina do. It should be like, what do I want to answer and what sequencer can I access? And that's going to be effective for people's projects that are quite diverse. Anybody have any questions about any of the sequencers? So, and this is just to highlight that, okay, if you're looking for mutations or, mutation detection, Illumina is kind of the established platform. The error rate's relatively low, you can sequence at depth, sequence fast. It's the one to probably focus on. If you're looking at RNA splice variants or translocations, Illumina long rate can get you there, but, you kind of have to be lucky. We'll talk about this a little bit. The PacBio may be more effective for that because PacBio is native sequencing. So you know exactly where the splice is because you see it happening. Novel RNA discovery, same thing. Illumina, PacBio. repetitive sequences, only the helicose or the sequel and the PacBio can see those very well. Everything else can't really see it that well. And then limited material, I think this is like, now everyone can do this because of these new amplification techniques that have developed. So, I'm going to move on to library construction. So, not all the sequences are the same, so guess what, library structure is not the same. I'm just using this as an example of like how do we do like single cell RNA-seq. So this is the original one that I had sort of adapted and used in circulating tumor cells, but basically it's a fairly straightforward concept. You have RNA, okay, and then what you had was you made a primer that had a bunch of T's, so oligo-DT, and you put a universal primer 1 sequence. So it's, again, an alien sequence that doesn't live in the genome. And you just have this primer, right? And so you use that for cDNA synthesis in your single cell, and you made DNA. And then you got rid of your primers, and then you got rid of your RNA with RNA stage. And then you do the same thing, terminal transferase, just like what I showed in the sequencing flow cell. So in this case, we used A's, so you put a bunch of A's here, okay? And then you come in with universal primer 2 that you sort of generate, and then you have an oligo-DT. And now it's not going to bind this side because RNA is gone, but it will bind this side. And then all of a sudden now you have 2 universal sequences on both ends of your cDNA. Okay, and so then you can amplify that using UP1, UP2 primers. And that's how you would generate your libraries. Okay, then you shear them. And these sequences are not unique to the sequencer. You shear them to a certain size because Illumina and the solid platform, the time solid doesn't exist anymore, likes to have things in the 200 to 300 base pair range for cluster formation. So in this case, we used ligation to put on your P1 and P2 adapters in the Illumina format. They call it P5, P7. So this is the newer one that most people are using. This is like Clone Tech, sort of single cell RNA-seq. Now they're on they call it HT now. It used to be V1, V2, V3, V4. It's just sort of the same iteration, I think. So it's the same thing. So you have again, sort of a universal primer that binds the poly A. And the main thing that's different here is they have this template switching thing that makes it more efficient in making cDNA and then coming back and making the other side. And so that's what most people are doing now. But conceptually it's the same thing. You have an oligo-DT, you come in, make your cDNA, get rid of the RNA, and then you come in and make the other side, and you have 2 universal sequences on the ends, okay? So that's fine if you're polyadenylated, right? But some of us are interested in the rest of the RNA world. So people are trying to do what we call total RNA-seq, and so this is Illumina's version of total RNA-seq. So first you get your RNA, so just your Kigen or your Trizol. Here, what they do is they do a couple things. So you can do the ribosomal depletion first. I mean, they may fragment first. But in any case, you have these little oligos that are directed against ribosomal RNA sequences, and they're typically biotinylated. And then the beads that you have are magnetic, and they have avidin. And so you mix them together, and then you are able to basically magnetically remove those ribosomal pieces. So this is a big variable that people don't think about when they submit to a core. That ribosomal RNA depletion, in every tube being 100% efficient is impossible. So your ability to remove ribosomal RNA is your largest variable that is occurring in this particular sequencing prep. And so that will affect downstream analysis, which we can highlight. But anyways, you shear this RNA, okay, so now the RNA is being fragmented. So this is not a great protocol if you have fragmented RNA to start with. You can't fragment fragmented RNA, otherwise it just gets more fragmented. But some people don't know that. So if you're the person doing it, and you know that your stuff is from FFPE, which is fragmented, you're going to skip the fragmentation step. But if you're someone at a core facility and you don't know, and you're like, oh, someone just told me to do total RNA-seq, they may do that because that's just written in the protocol. And so you may get no sequencing because you just forgot to tell them that it's already fragmented. It's just like things to be aware of. That's why I tell people like it's better just to do your own library construction. So my lab does everything from RNA extraction all the way through because I spent my postdoc working on that. Anyway, so after you do that, you do cDNA synthesis, second strand, and then in this case you put a 1A at the end. And then they use that as a way to do a ligation of P5. So if you have this overhang of a single A, there's a particular type of ligation used. And so P5 is again, that thing on the Illumina is for the sequencing flow cell. P7 is on the other side for the cluster. And so in their case, they developed this as stranded, basically because the second strand incorporates U's. And so when you try to do PCR amplify, the U's don't work. And so that's why you maintain stranded information. Does that make sense? Because only one side is being amplified. Any questions about that? So the biggest thing here is that can kill you. All right. So there are other total RNA-seqs out there. I'm starting to like this one. This is by CloneTech. It's the same kind of concept of getting rid of ribosomal RNA. This didn't come out very well. But how does this one do it? It doesn't use beads. It uses RNase H. And does everyone know what RNase H does? Anybody? RNase H. I kind of was talking about it before. So RNase H degrades RNA, but only one type of RNA. You guys know? Is that funny? Okay. It degrades RNA that's complex to DNA. So RNA-DNA hybrids, it takes out the other side. That's how you do, so if you ever do cDNA synthesis with superscript 3, there's this thing called RNase that you add for second strand synthesis. You have to get rid of that other RNA on the other side before you complete the second strand. So this one, instead of using those beads, it uses an enzyme to degrade any kind of RNA that is bound to basically DNA probes against ribosomal RNA. Okay? And so it's a different way of depleting ribosomal RNA in your sample. And so this is something that you should be aware of because this one may be a little more efficient than the other one, which is bead-based. It's like mechanical. I'm not sure if that's actually true, but this particular technique you can do with small amounts of RNA. So the bead-based one, you need a lot of RNA because you have to add these beads to some volume, and then so you can't really do total RNA-seq off that protocol. I think you need a minimum of 100 nanograms of RNA. But sometimes you just have a tiny biopsy from a patient, or you have a small amount of material for whatever reason. You can't get away with that. So this kit is designed to deal with small amounts of material. And it also gets rid of the RNA, the ribosomal RNA in a different way. Okay? All right, so those are just examples. This is just a very busy slide, and I don't want to get into all these things, but I think the biggest thing to understand, when you say RNA-seq, that could mean a lot of different things. Is it mRNA-seq? Is it total RNA-seq? If it's mRNA-seq, you have biases in sequencing. It's usually 3' biased, right? So you may not see, let's say, one question I always get when I do RNA sequencing for, pancreas cancer is, oh, can you see the KRAS mutation in your RNA? And I'm like, I wish I could, but single cell RNA-seq is 3' biased because you do an oligo-DT thing, right? And so, what's the problem with that? KRAS is an exon, I think, 2, right? So it's on the 5' side of the RNA, so you almost get no reads up there because the cDNA synthesis is not efficient. That's something just to be aware of. It's if you do an oligo-T, sometimes it's good for counting, but you may not be able to see, RNA, DNA events or RNA editing, as some people call it. So, that's why people want to do total RNA-seq. But total RNA-seq is very difficult to do in single cells, which is because, you have so much other ribosomal RNA, you have DNA, and that's why most people have not done single cell total RNA-seq. I think it's technically difficult, but potentially doable. And you just have to be aware that whatever prep you do, I think, again, it's always better that you know what you did, because then downstream you'll be able to be like, oh, you know, this is a variable that may be affecting my data analysis right now. When you send it to the core, they just do what's on the menu. And so sometimes it's not as useful. So I'm not saying anything bad about the cores, but I'm just saying if you're doing this class, you probably want to know what your data is, and I would say you should definitely actually do library construction so you know exactly what happens. Because only you will know, you know, this sample may have gotten something that it shouldn't have gotten. And only you will write that down on it in your notebook. The core facility is not going to do that. They're like, hey, everything's sequenced. Right? I'm just being totally practical here. Okay, so I'm just not going to go into this. So let's say you're interested in like splice variants, like ARV7 is the big lung prostate cancer. There's the EGFRB3 splice variant and glioblastoma. Can you see that? So if you were just to do Illumina, you would have like basically 100 or 150 base pairs on this side, 150 on this side with pair-end sequencing, and then about 100 or 150 in the middle as not sequence. And so let's say this is exon 1 or gene 1 and gene 2 for a translocation. Let's just say it's translocation. If you sequence here, you would be perfect, right? You would, oh, I nailed it. It's like a line like this. But guess what? When you run this read onto your aligner, it's not going to align. It's only going to align partially to this gene, but it doesn't know to do partial alignment. So even though you have this read in your data, you can't actually see it unless you tell the program to align partial reads. You would have to kind of guess, like, align the first 10 bases to anywhere in the genome, the next 10 bases in the other part of the genome. So people think that, oh, this would be perfect, but it's actually not. Because computationally, this is very difficult. This is actually the perfect read. You have read 1 on one gene and read 2 on another gene. But that's like, you gotta be really lucky. Right? You gotta nail the transcript in a 300 base pair window and hit it right there. And then read 3 you totally miss, and read 4 you totally miss because it's only on one gene. Okay, so that's why people have been doing PacBio. Because PacBio would, if you took, if your translocation was at some level, at a higher level, you would actually see the transition when you looked at the read, because the read is so long. So you would be able to see, oh, this is the, this is the N-track gene, and then all of a sudden it turned into a different gene, because it's all in cis with each other. Does that make sense? So a lot of people are doing the combination of the two. So you're trying to say, okay, I want to see what genes there are with Illumina, and then I'm going to see if I see any funny rearrangements using my PacBio. And so this is what some of the splice variant people are finding for novel identification or translocations. Okay? Any questions? All right. DNA, RNA library purification. So I just showed this just briefly, but again, this is magnetic bead based. And so a lot of times you can add different amounts of beads in to purify different sizes of DNA. Okay? So the more beads you put in, the more basically ethanol or isopropanol goes in with it, because that's what the beads are mixed in. And so that creates more sort of stringency, which then drives those DNA to the beads. So if you're at like 0.5x SPRI, you would basically find high molecular weight stuff above 500 base pairs. And as you increase, you would get like lower molecular weight stuff. So usually when library construction, you try to grab this region here between 500 and 200 base pairs. And so a common technique we use in the lab is called double SPRI. So you would actually put 0.5x in, this high molecular weight stuck to the bead. Supernate's got the low molecular weight stuff. You take that and you put it in a different tube, and you get rid of the high molecular weight stuff. And then you take that supername and add 1.0x or 1.8x. I think it's 1.8 is what they usually do. And now you've gotten rid of the top stuff, but you still combine the bottom stuff and purify that away from primers. So these are these like little tricks that we do to change sort of RNA or DNA extraction, but also libraries for sequencing that aren't necessarily written in the book. So for the ChIP-seq people, sometimes they don't like to use those beads because the amount of chip material they have is so tiny. So there's this like machine, it's fairly straightforward, that is in the Scioto lab that people can use. But it's called Pippin prep. I think it's still there. It's basically agarose. It's just like running gel electrophoresis. And then you run a ladder next to it. And then it's got this little sipper underneath the gel. And so when your sample gets to a certain size as it's running based on the ladder, you can tell the electric field to go down. And so it will move the DNA this way, and then it's like 2D electrophoresis, right? And then it will actually force the DNA into your sipper, and now you can purify it this way. So this is like the fancier way of cutting out the gel, except you don't need ethidium bromide, which is an intercalator and affects your sequencing. And the second thing is that you don't have to cut the gel. So it's pretty cool, and I think the ChIP-seq people like this a lot. Okay. Can you make your own custom library? So this is the short answer is yes. This is the sequence. So this sequence is P5. This is P7. So you need those at the ends to create clusters. And then this red sequence is index sequencer read 2, and this is read 1. So what are those? In order to initiate sequencing, you actually have to prime with a primer. That's why when you send it for Sanger sequencing, you all send the primer with your piece of DNA that you want to sequence. So these red and blue ones are the things that the Illumina sequencer will add when it's on the flow cells to initiate sequencing. So the actual sequencing doesn't start from P5 or P7. It actually starts here after read 1, which is basically the sequencing primer. But anyways, you can actually make your own custom Illumina libraries. Like let's say you wanted to make P5, P7, or a P5 primer with read 1, and then you wanted a cDNA with a specific sequence, you would make a really long primer for that. And then create basically a library with the Illumina adapters as well as the sequencing read primer sequence within your library. Does that make sense? It's very straightforward. So why do we care about sequencing quality? So this is quality reads. Did you already do this quality read?
A little bit yesterday, but
yeah.
you can, yeah.
All right, the bottom line is like Fred score is like your quality sequencing. So on average, it's about a Fred's quality score of 30, which is a 99.9% accuracy, so 0.1% error, right? And so this is a typical read from single cell data. And so you'll see in this sample here, this is green is basically a good Fred score. And as the sequencing goes on from the beginning of the sequencing to the end, so this is about 100 base pairs, it usually does this kind of thing. It sort of slows down. So this one looks good. But this one goes from good to terrible. Right? So your errors are like getting worse as the sequencing goes on. And that's a known fact. Okay, at the ends of the sequencing read, your quality is worse. You'd be like, well, why do I care? Well, you may have like not a great library, and like let's say like 50% of your reads don't align. It could just be because you have crappy stuff at the end. And so all you have to do is computationally chop off 10 base pairs and realign, and oh look, it works. Okay, so those are the like little things to be aware of. But I think the bigger things is, and this is what I sort of just talked about. So there's this big paper in this little journal named Science where they sequence RNA and DNA from the same thing. And they said, oh, look, there's so many differences in the RNA and the DNA from the sequences. Okay, and what they found, and this is a big lab, so it's not this small. They found like thousands of these things, A to G events, T to C. So this was evidence of RNA editing. So it's a difference that you would see in the RNA that was not in the DNA, and it looks like a mutation in the RNA. And they even showed that some of them, creates mutated proteins that lead to a functional protein change. All right, so that's pretty cool. And this does happen, right? But does it happen to this extent is the question. And that's why it was in science. Okay. So it turns out that there's like 3 different labs that came out a year later and said, maybe you're not right. And so they sent letters in because it's science and they had some issues with it. Guess where all those RNA events were occurring? The end of the sequencing run. They're all happening at the end. And so this is one end, and this is the other end. So this is your mean fraction of events. So basically, these 3 independent groups found out that, oh, your RNA DNA events are mostly sequencing errors. Oops. So, you don't want to be that postdoc or grad student or med student or whatever. Okay. All right. So that's just to say, okay, you got to be aware that like that thing I told you about the quality score is like, most people don't really think about it. You're just like, oh, my computational guy is just gonna know what to do. Well, if you're trying to look for mutations, just be aware, like, if it looks, okay, what I tell all my folks, if it looks like too good to be true, it's probably not true. Okay, that's all I'd say. All right, so additional reason to have some caution with mutation detection is that, how many pipelines do you think there are for mutation detection? There's a lot. This is a Nature comp paper. I thought this paper was amazing. But what they did is they did every combination of all the aligners that you can think of for mutation calling. So they did like SSM calls were done with these, and they did different aligners, and then they had different detection software callers. Okay, and this is true positive. This is false positive. Look, this is the same data. What are the false positives for this combination? And this is mutat, which is the original one that wrote 6,300 mistakes. Right? So what if you chose the wrong combination just because someone else was doing it and that's what everyone was doing? My point is that just be aware that you think that these tools in R are like foolproof, but they're not. They're not. They're freeware. So just be aware that some of them might not work that good. And you don't want to be someone that's like basing your entire postdoc or your grad student or whatever you want on something that is has 6,000 events that could be wrong, right? I mean, it's pretty scary. Any questions about that? Definitely consult your computational. Okay. So these are just like the summary points. I think I actually have more slides. It's not actually a summary, but my point of showing all of this is like there's a lot of variation in sequencing. Sequencing is not all the same. Just sequences are different. Like instructions different. Your questions are different. Computational pipelines are different. Error rates are different. Just be aware that this is not just a machine that's a magical machine that gives you perfect data every time. All right, I'm going to end with this normalization thing because I'm an RNA person, and this is the one thing that I find most disturbing for people. So, we had done the single cell RNA-seq, which I already explained, and this is in circulating tumor cells, this is the thing that I do. But what we learned from single cell RNA-seq a long time ago, before everyone was doing it, was, like if you take 5,000, so there's about 10 picograms in one cell for RNA. You take 5,000 picograms, so that's what, 500 cells equivalent, and you run it on the same sequencer run. You know, the alignment's pretty tight, so each one of these dots is a gene, okay? So, you know, it should be perfect, right? It should be a straight line. But as you get lower and lower, so this is 500 picograms for 50 cells, 50 is 5 cells, 1 cell, most of the genes that you're seeing are not correlating. And this is because of detection issues. So sometimes you detect it, sometimes you don't because it's rare. Because at 1 cell, you just don't have a lot of material. So that's a problem, right? Because that means like a lot of your variation that you may find by differential expression can be just because you can't see it, not because it's biologically real. Does that make sense? All right, and we did this in our own data, and it looks equally as terrible. This is single cell data. This is single cell data. This is actually just RNA, spiked RNA. So this is 10 picograms RNA, 10 picograms RNA. It's not because it's single cell, it's because the amount of RNA in there is so low that sometimes it amplifies and sometimes it doesn't. Okay. All right, so what does this cause is that if you look at different types of cells, so this is white cells, this is a cancer cell line, this is a non-cancer cell line, these are tumors, these are circulating tumor cells, and you look at all the potential genes in the genome that are expressed as RNAs, or coding RNAs, you know, there's a high variability of gene detection, because not all cells are producing all the genes in the genome, right? There's 30,000 genes, so some things are only using 5,000 genes, some of them 10,000. So white cells, you know, most of those reads, most of the genes were not seen. So 90% of genes were 0. Okay? And so what if you were to do a differential expression between white cells and cancers? What do you think all those differential expressed genes are going to be? They're going to be all these things that are 0 in white cells and something in cancer cells. And it's not because it's biologically meaningful. It's just that that cell type doesn't produce the same number of genes as another cell type. So all you're seeing, the differences are, is your white cell and your cancer cell, and you're not really seeing those things. So why is that important? So as you compare samples that you want to compare, so white cells and cancer cells, who cares? But cancer cell from a tumor compared to a CTC, the number of zeros starts to matter. You're like, oh, this is a tumor cell, this is a tumor cell. These are biological differences. Guess what? It's not a biological difference. It's just that, you know, these cells we're seeing more zeros than the other. It could be biologically relevant, but you know, is 5 transcripts in a tumor cell in the primary tumor compared to 0 significant? To you, maybe not, but to DEC it is. And this is just showing, like, this is how this data looks. If you just like cluster it, you see these what we call like bursty things, because these cells have a lot of expression here, but they don't have a lot of expression in anything else. So when you normalize the data and you have less genes, you know, it looks like the amount of gene expression is so high. But it's just because the denominator is so much lower. Does that make sense? And this is just to show, you know, when we sort of corrected for all those zeros with different ways of normalization, it actually worked. So I wanted to skip this part. This is just showing that if you change how you normalize the data, you can cluster and this is led to the paper. So you know, let's say you want to do differential expression now. There's like all these pipelines that you're learning, like DE-seq2. Do you do DE-seq2 and
we'll do it later on. Yeah.
EdgeR or no?
We'll do DE-seq2. We'll cover EdgeR as well a little bit.
Right. So single cells, yeah, we're all interested in single cells because this is what DE-seq2 did. Okay. So these are all the genes that are differentially expressed between the primary and the CTCs. Circulating tumor cells. So I always tell my people in the lab, like, okay, even though DE-seq says it's statistically significant, just heat map it. Because if it doesn't look right, it's not right. So this is what DE-seq called as differentially expressed. I don't know. Do you guys see any differences? It's really nothing, right? It's like nothing different. I mean, maybe this jump out here, but it's really nothing different. This computational pipeline call this differentially expressed. There's like absolutely nothing here. right? Like if you were to send this for publication, people like, you're kidding, right? So this is just a, an illustration that pipelines are not perfect. People think, oh, it's going to give me an answer, but you actually should just look at the data. If it looks right, then you know the pipeline's working. If it doesn't look right, it's probably doing some normalization that's not good for your particular data. These are just all these examples that Ben Winter had basically showed nothing's different. Okay, so yeah. Okay, so then EdgeR is the other big one that people use, and maybe this is even worse. So these are all the differentially expressed genes. And you can see that it's really not, right? It's really not. And so when you did a formal t-test, there's nothing there, but EdgeR was spitting these things out. So, and then if you just did a straight t-test, student t-test, then you started seeing things that were different, okay? And so I show this because, just to say that, like, sometimes the simplest statistic is better than the fancy EdgeR DEC, because, there's a reason why people have used the t-test for centuries or however long it's been. It works pretty well sometimes, and so only when it doesn't work is that I would say then start to think about different normalization methods. So this is just showing that these genes are high and these genes are low, and they sort of look like. And this is just more than zero. So one of the ways that you can also do differential expression is not like what we call sort of the standard t-test. T-test is like a gene that's 5,000 versus 500 is different. But another way of doing it is like, well, what if you just did it by rank? So if this gene is always the highest in each of your single cells, it would be worth more. And so this could be 5,000, this could be 2,000, this could be 3,000. But it's, that variability of 5,000 to 2,000, 3,000 is not incorporated in the statistic, but it's what we call a rank product or a non-parametric or way of doing it. So this is, a way of doing it. So what you do is you just multiply these ranks, and you can get a rank score. And so this takes into account where is your gene expressed relative to other genes, and not so much what the absolute RPM values are. And so this is sometimes useful when you're dealing with very noisy data where there's a lot of variability in gene expression. Okay. All right. And this is just showing that the t-test looks better when you do this, or the rank product looks good in separating these two population limits. So, and this is what we ended up using this rank product to identify differentially expressed genes in single cell CTCs compared to other types. And so that's pretty much what I wanted to just highlight, because I don't want to go over time. But normalization of your data is, at least in RNA-seq, is a huge thing. So if your denominator is very different, and so that could be, some of it could be some of these artifacts in single cell data, where some cells have lots of zeros and other cells don't. Some of them could be because of your library construction. So let's say your ribosomal depletion is not great, okay? And so this one has 50%, this one has 10%, and you're interested in non-coding RNA, so you align to the entire genome, right? Well, all those ribosomal reads will align to the genome because they're in the genome. And so if you didn't assume that the ribosomal reads should be removed because that's what you were intending to do with the ribosomal depletion, you have different amounts of ribosomal RNA in your denominator that is not what you wanted to intend. But because you're looking at whole genome alignments, if you didn't tell your computational friend or you yourself did not say, oh, I need to digitally subtract anything that's a ribosomal RNA because the sequencing prep was intended to remove all of it, then your normalization is different, right? So your differential expression is going to be dictated by how much ribosomal RNA contamination you have, and not so much about what's actually biologically relevant. Does that make sense? So always think about normalization. Sometimes simple normalization is better than doing fancy DEC or EdgeR. Just because it sounds fancier doesn't mean it's better. I learned that from my mom. So, and then the one other thing I want to tell people is that anything you do, you will find a p-value. You will find a p-value because a p-value is just a probability. And so a lot of people are like, send their sequencing data off in omics and they pray that the p-value will be like negative, 1 to 100, negative 100, and there's like one gene and it's going to be the clear answer for the cure for cancer, but that never happens. So you will always get a list of probably 300, I'll say 300 because I feel like that's the number that I will see. 300 genes with some kind of p-value of significance. And if you could clone yourself 300 times, then you'd be set, but you can't. So just be aware that sequencing data just creates more questions than answers, usually. And so I always tell people, always have the question before you do the sequencing. Because if you go in and just think that the sequencing is going to give you the answer, you're not going to find it. But if you go in with the, this hypothesis thing, and you look in the data for the hypothesis to be true, then you hopefully are right, but sometimes you're wrong. But that's the better way to approach it. So always try to think with a question and not sequence, just a sequence, hoping that you will get the great idea that everyone's been looking for. Because I can guarantee you a lot of people have done that and they haven't found the magical egg. Okay? And that's all I got. So any questions before I have to leave? Does that make sense?
All right. Well, thank you so much.